Alright, so in this video we're going to talk about different types of peripheral motor endings. If you remember, we talked about how there's two different branches of the motor division of the peripheral nervous system. We said that there was somatic motor nerves and autonomic motor nerves. So we're going to first focus on the somatic motor nerve ending here, which innervates skeletal muscle, whereas the autonomic motor nerves innervate visceral muscle and glands. So in terms of skeletal muscle innervation, this takes place at what we call the neuromuscular junction. We talked about this back in chapter 9, and we said that the major neurotransmitter of this was acetylcholine, and it's released when the nerve impulse reaches the axon terminal. And once acetylcholine enters a synaptic cleft, it binds to receptors that result in the movement of sodium and potassium. This leads to a net depolarization of the muscle cell, which we called end-plate potential. This end-plate potential spread to adjacent areas, which can trigger the opening of voltage-gated sodium channels. And if that happened, then we had an action potential that spread down the T-tubules and eventually led to a muscle contraction. So to summarize this process, we talked about how this is the point of communication, which is the neuromuscular junction. So we see our somatic motor nerve axon here, innervating in an individual skeletal muscle cell. And if you zoom in at this point of contact, this is the neuromuscular junction. So once the action potential arrives at the nerve terminal, that triggered the opening of voltage-gated calcium channels that allowed for a calcium influx. That influx of calcium then triggered the exocytosis of neurotransmitter-containing vesicles because they fuse to the membrane here, the presynaptic membrane, causing ACH to flood the synaptic cleft. Once in the synaptic cleft, that acetylcholine here could bind to nicotinic receptors or acetylcholine receptors, and once bound, these receptors open up this ligand-gated ion channel. We have a net influx of sodium and some efflux of potassium, but that net influx of sodium, if with enough of it, caused end-plate potential, which could spread to nearby patches of membrane that may trigger an action potential. And we said what stops the whole process was either stopping action potentials or having this enzyme acetylcholinesterase come in here and break down the neurotransmitter to remove that signal. Now, this differs from autonomic muscle because autonomic motor nerve endings and visceral effectors are more simple than somatic junctions. We see that at the autonomic nerve endings, branches form synapses en passant, which means synapses in passing. And these actually occur at what we call varicosities. Now, acetylcholine and norepinephrine act indirectly via second messengers rather than ligand-gated ion channels, and visceral motor responses are more slow to react than the somatic motor responses. So what we find here is our autonomic nerve ending, and you see that at these little blebs, these are the varicosities. Now, these varicosities are full of mitochondria, which can first of all make ATP to power the exocytosis of these neurotransmitter vesicles. And these vesicles actually then, exocytose directly onto the effector muscle here. And this example is showing smooth muscle contraction. So this could be like a parasympathetic nerve, like let's say vagus nerve endings, that actually then spread out onto the smooth muscle of your digestive tract, and this might actually cause like contraction of your digestive muscle. But remember, these responses in the autonomic muscle, like smooth muscle, cardiac muscle, and glands, they're actually more slow than the somatic responses.